This is the renal corpuscle. The main thing in the renal corpuscle that we're concerned about is that it contains a capillary bed. And this capillary bed is a little bit different than the ones that are in your body. And so we give it a specific name called the glomerulus. And so this glomerulus, it's just a capillary bed. And the capillary type is a fenestrated capillary. We talked about 3 different types of capillaries in chapter 21, and fenestrated was just, it was like middle. It was like a little bit leaky, but not leaky enough. I've also cut open the capillary so you can see some of the things inside of it. So there's 3 little windows there that allow you to look into it. So just don't forget, this capillary bed gets a specific name called the glomerulus. I've got some red blood cells in there. I've got some white blood cells in there. I've got big toxins, like maybe an antibiotic or something like that. And I've got albumin. I have a couple of other things there, but we'll talk about them in a little bit. This capillary bed, the glomerulus, is fed by an arteriole, and that's like any other capillary bed. So we call that arteriole the afferent arteriole. And yes, it brings blood from the heart to this capillary bed. So it's whole blood, plasma, buffy coat, and red blood cells. This is what makes the glomerulus different than any other capillary bed in the body, is that it is not drained by a venule. It is drained by what we call the efferent arteriole. And that arteriole brings the filtered blood to another capillary bed. And this is what makes the kidneys capillary beds so unique, is that they do the process in 2 parts. They have 2 capillary beds. Like you have to go through A and then you have to go through B. So this is what makes the glomerulus very different. It's drained by an arteriole that we call efferent. This capillary bed is sunken into a serous membrane. And your book had a really great picture in like chapter 1 where they had a balloon and somebody was like punching the balloon, and the balloon rose up around their fist. And that's what we have here. This capillary bed is like the fist, and it's punched into a balloon. The outer layer of the balloon is called the parietal layer of the renal capsule. And what you can see is that it's made up of simple squamous epithelia cells. Now, you want me to draw the green line from that parietal, like just all around that glomerulus there, right? Well, it doesn't actually work like that. The visceral layer of the renal capsule is this specialized layer of cells called podocytes. And podo means foot. So these podocytes, what they do is they have like little toes that reach really far and intermingle with the next podocyte next to them. And it forms this secondary filter. So if you're in the glomerulus, you have to be able to get through one of those fenestrations, and then you have to be able to get through the toes, the interlacing toes of the podocytes. So these podocytes are the visceral layer. I forgot to show you there that the parietal layer extends into the tubule, and so it becomes the proximal convoluted tubule. Just know the anatomy of this picture. We'll put it all together in a moment. The visceral layer, the podocytes, and the fenestrated capillary are what your book and other books like to call the filtration membrane, right? So you have thousands of these renal corpuscles in your kidney, and all of their little visceral layer and fenestrated capillary together is the filtration membrane. So we have the glomerulus and the podocytes making up that membrane. Between the parietal and visceral layer is what is called the capsular space. This sometimes is called Bowman's capsule. Sometimes they can call it the renal space. I don't like that term, but capsular space. And it's full of filtrate. You wanted me to say urine, but urine is what is produced as we drip from the renal papilla. This area here, filtrate, is not ready yet to leave the body. It needs to go through some adjusting in that second capillary bed. These little cells, do you see the orange little cells? They're like little bumps. They're all around some portions of the glomerulus. These are called mesangial cells. Your book, many books, they'll tell you they do lots of different things, and they do. They secrete fibers that help the fenestrated capillary stay intact. They're phagocytes, so they clean up any debris in the area. I've seen some books that say they maintain the general health of the glomerulus and the podocytes. Okay, yeah, they do. Over here, outside of the glomerulus, that's what over here on the left-hand side is. There are some cells lining the glomerulus on the outside that are called extraglomerular mesangial cells. So we have the mesangial cells on the inside, and then we have mesangial cells on the outside as well. The extraglomerular mesangial cells, we'll find they function a lot in communication. They are not neural cells. They're paracrine cells, so that they do secrete hormone-like chemicals, but they don't enter the blood. They go like 2 inches. The other kind of cells that are here that I have labeled are called the macula densa, and those are chemoreceptors, and they're in the distal convoluted tubule. So you have to remember that the nephron is not all stretched out like when we make drawings in the next slide. The nephron twists back on itself so that this DCT can come into contact with the glomerulus. It's a feedback system. Oh, I'm making you this filtrate. Oh, you know, what you made isn't exactly right, so speed it up, slow it down. There are smooth muscle cells around the afferent arteriole. There are also some around the efferent. So this lets you know that we can constrict the afferent arteriole if we want to or need to. And there are also these cells called granular cells that are in the afferent arteriole. And those are basically, they're mechanoreceptors, they're baroreceptors, they're stretch receptors. So these receptors here, when the afferent arteriole stretches due to low blood pressure, these cells have a feedback mechanism and they cause the afferent arterioles to constrict. So all of these structures here are highly specialized for the first capillary bed in the renal corpuscle. What we're going to learn is that the mesangial cells, the macula densa, the granular, that all those very specific cells in there form a feedback mechanism that allows the kidney to control itself despite anything else that's going on in the body. So basically the kidney gives the finger to everything else in the body and says, no, I'm doing this and I don't care if it's damaging. I just think of the kidney like an insolent child, like, no, I'm doing this. The last thing that I want you to notice is that in the afferent arteriole, we have albumin, toxins, blood cells, but we also have ions, sodium and potassium. And that little hexagon there, blue hexagon, is glucose. Notice how those things are not in the efferent arteriole. It seems that those things go to the second capillary bed in a different way than big toxins, albumin, white blood cells, red blood cells. Take a look at the capsular space. That's where glucose and sodium and potassium end up. Those three things are able to fit through the fenestrations in the glomerulus, and they're able to fit between the toes of the podocytes of the visceral layer. Those substances are small, and they enter the filtrate. The other things, the cells, albumin, and toxins, are large in size, and they don't fit through the filtration membrane. So those things stay in the efferent arteriole. What's happening here is we're separating substances in the whole blood by size. We're saying to all the small stuff, you go this way in the filtrate. And we're saying to all the big stuff, no, you stay in the glomerulus. What's the dividing line? How do you know what's big and small? I mean, the kidney knows, obviously, but how do we know? Albumin. Albumin is a protein that's in your blood. Albumin is the smallest substance that doesn't get filtered. If you're bigger than albumin, you're going to stay in the glomerulus. If you're smaller than albumin, you're going to enter into the filtrate in the capsular space. Oh, I'm sorry. I meant to say these cells over here that we talked about, they form a structure that textbooks call the juxtaglomerular apparatus. So when it refers to the JGA or the JGC, some websites will call it the juxtaglomerular complex. It's talking about all of these sensory cells that feed their information directly to the glomerulus. These chemoreceptors, mechanoreceptors, they're senses don't go to the brain. They go right to the glomerulus. This is one of the pictures that you have to be really careful of because this is not what a nephron looks like. This would be a good picture of a nephron if we were to take that loop there in the center and just fold it over. So remember, nephrons are not stretched out like this, but this is how we put them in a schematic so that we can understand the flow in a nephron and kind of read the picture from left to right. I've already highlighted on this nephron a blood capillary. And remember that blood capillary has like 2 capillary beds in it, one up there at the glomerulus, and then another one that runs around and through the nephron. So that capillary there being all stretched out, that's not how it is. It winds itself around the nephron. Don't forget, let's take a moment. Let's think where we are. We're in a lobule of a kidney, right? We have the cortex above us where the cortical radiate arteries and veins are squiggling out to the edge. And then we have the medulla, which is the juicy center of the kidney. I'm going to give you a hint right now that we're going to talk about next week. The medulla is very concentrated in salts. Just FYI, keep that in mind. Okay, now whole blood is going to enter from the cortical radiate artery, and whole blood goes into the afferent arteriole. That leads us to the glomerulus. And if things don't get filtered into the glomerulus, they will stay in the efferent arteriole, and they'll make their way to the peritubular capillary bed, or a capillary bed that's called the vasa recta. And you're saying to me, how do I know which one? Well, if it's a nephron that's only in the cortex, it's a peritubular capillary. That's not this nephron. If it's a nephron that dips down into the medulla, that's what one of these nephrons are, then it's a vasa recta. So different placement, different nephron, different capillary bed. Our glomerulus is still the same. It's just the second capillary bed that's a little bit different. Once once the capillary finishes like twisting around the nephron, the blood is taken up by the cortical radiate vein and thus delivered back to the arcuate, interlobular, segmental, and then back to the renal vein. So the peritubular capillary bed and the vasa recta are normal capillary beds. They are fed by an arteriole. They're drained by a vein. Take a look at the glomerulus here. I actually do have spaces, fenestrations in that capillary that dips into the glomerulus. I have the glomerulus here highlighted in orange. You can see the simple squamous cells of the parietal layer and the teeth or the feet of the podocytes. Okay, that capsular space there fills up with filtrate, and filtrate will then run into the PCT, or the proximal convoluted tubule. I'm sorry, that gray circle there this is the renal corpuscle. That's what the gray circle is. The proximal convoluted tubule, it's not straight like this, it bends and twists, and it's covered with cuboidal cells, simple cuboidal, that have lots of microvilli on them. Filtrate then descends through the descending nephron loop. This was once called the loop of Henle, so you might see that. This loop has simple squamous cells on it. After the filtrate descends the loop, descending into the cortex, the filtrate then ascends the nephron loop. The ascending nephron loop is covered in simple cuboidal cells. Notice that both arms of the loop have a thick portion and a thin portion. And that's true. The nephron loop deep into it, where it dips into the medulla, is thin. But the nephron loop where it is in the cortex, it's actually quite thick. This area here is called the DCT, or the distal convoluted tubule. It has cuboidal cells on it as well. And remember, this is the part of the nephron that twists back around to come into contact with the glomerulus. So again, this picture is not anatomically correct, but it allows us to lead from left to right. The last thing is that we dump all of our filtrate out into this collecting duct. And the collecting duct has different types of cells in it called intercalated and principal cells. The collecting duct does not just drain one nephron. It drains more than one. It drains many, many nephrons. The collecting ducts all converge at the renal papilla. And at the renal papilla, the collecting ducts drip urine into the minor calyces. At this point, you can call it urine. It's ready to leave the body. We call it filtrate when it's in the nephron loops or the convoluted tubules, and that's because it's not ready. It has some adjustments that need to be made. Of course, as the filtrate flows along the PCT, the nephron loop, and the DCT, it is constantly exchanging with that capillary. Things are constantly moving out of the nephron loop, out of the nephron, and things are constantly moving into the nephron. The things that move into the nephron from the capillary into the nephron are things that you will secrete in your urine. And so that process of moving something from the capillary to the nephron is called secretion. That's the process of saying, I don't want this, get rid of it. There's also this process of reabsorption, and that's the one that's a little bit more important. That's what takes things back from the PCT, the nephron loop, and the DCT. In reabsorption, we're saying, oh, did you filter this stuff and designate it to leave? No. I'm going to take back all the glucose, almost all the water, and pretty much all the ions right away. And then we're going to take back more water. And then we're going to take back some salt. And then we're going to take back a whole bunch of stuff. The collecting duct does like the final adjustments so that urine can leave the body. I just want to put out that secretion and reabsorption will change depending on what is in the filtrate and depending on what is in the capillary bed. So if your blood glucose levels change or go high, you might find glucose in the tubule. There's only so much that you can reabsorb. If your sodium levels are high, you might find more sodium in the tubule. You should always find potassium in the tubule. Your body never really likes to keep it. It likes to secrete potassium as much as it can. We will, in a future week, talk about the specifics of that movement pattern, the secretion and reabsorption into each one of these structures. But right now, it's important to understand the anatomy because we're going to start using all of these terms like you know exactly what they mean.